Assalamualaikum and good morning. I am Isrin and I will be teaching you glycolysis and gluconeogenesis. The main learning objective today is first is to be able to understand the role of glycolysis in carbohydrate metabolism. Second, to be able to explain the steps and reaction of glycolysis and the respective enzyme that involve in the roles of glycolysis. So for the outline for today's lectures will be the overall pathways of glycolysis. And these pathways are basically divided into two stages. One is the conversion of 6-carbon glucose to 3-carbon glyceraldehyde triphosphate, and the conversion of glyceraldehyde triphosphate to pyruvate, and also the anaerobic metabolism of pyruvate and the energy production, which is ATP in glycolysis. So before we learn about the pathways, you need to know about directions, anabolic reactions and catabolic reactions. So anabolic reactions is a reaction to form a complex from a simple precursor. For example, here the synthesis of polysaccharide or glycogen from glucose. And in this reaction, energy is required. So this is an endogenic process. And then the energy is provided by the breaking down of ATP to ADP and inorganic phosphate. And for catabolic reactions, is the breaking down of complex molecules like proteins, polysaccharide, lipids to the simpler molecule like like carbon dioxide, ammonium, and water molecules. So this is some terms that you need to know and be familiar with. And we have this NAD plus or NADH. So this is one is oxidized form and one is the reduced form. And then their full name is nicotinamide adenine dinucleotide. And you need also to know about FADH, FADH2 in the oxidized and reduced form, which is flavin adenine dinucleotide. And AMP, adenosine monophosphate, ADP, adenosine diphosphate, and ATP, adenosine triphosphate. Okay, what is carbohydrate and how carbohydrate in the body? So carbohydrates is the fuel for activity, and then the simplest, and then carbohydrate sugar is glucose. And glucose is stored as glycogen in animals and starch in plants. And glycogen, when glucose is when glucose molecule are abundant, so glycogen is made and stored in the liver when blood glucose is available and ATP are abundant. And the liver stored 1/3 of the body's total glycogen, and it will be released when it is needed, meaning if you need glucose. And the other 2/3 are stored in the muscle cell, where it is used mostly during exercise. Glucose is metabolized through cellular respiration. So what is cellular respiration? Is a reaction that break down carbohydrates, fats and proteins in the presence of oxygen to produce carbon dioxide, water and energy in the form of ATP. Glucose is metabolized through cellular respiration, which involve 4 stages. One is glycolysis. 2 is acetyl-CoA formation. And third is the citric acid cycle, which is Krebs cycle or TCA cycle. And fourth is the electron transport chain, also known as oxidative phosphorylation, ETC. What is glycolysis? It plays a key role in in a way organism extract energy from nutrients. So you need glycolysis pathways to extract ATP from glucose. So this is the first stage of carbohydrate metabolism. And then this pathway itself, it can happen in the presence or absence of oxygen. It doesn't matter. So glycolysis, it happen in cytoplasm of the cell. So you need to know in a cell um, what is the pathways happen in cell, and some of the pathways can happen in the uh, cytoplasms. Okay, so basically for glycolysis, uh, these pathways happen or occur in the cytoplasm of the cell. Okay, basically in these pathways, so glucose is a 6 carbon molecule. So it needs to be breaks down into 3 carbon molecule. So from 6 carbon glucose molecule is converted to 3 carbon pyruvate molecule. So when this happens, the energy is released from one molecule of glucose in you and is used to make 2 ATP and 2 NADH molecule. So basically, this is the overall um, what will happen in glycolysis. So we are going to look into it further later on. And then towards the end of glycolysis, pyruvate is formed. And then we are going to look into what happened to this pyruvate, which is the fates of pyruvates. And this is glucose molecule. You can see glucose has 6 carbon molecule. So it will produce 2 ATP electrons, and at the end it will produce 2 pyruvate molecules from one molecule of glucose. So in general, okay, this is the glycolysis pathways. Okay, so here is the glucose. Okay, from glucose, this is the pathways of glycolysis. At the end, it produces 2 pyruvate. Okay, so this pyruvate, it can goes into anaerobic glycolysis or aerobic oxidation or anaerobic alcoholic fermentation. So from these 3 stages, okay, towards the end, it will produce a lots of ATP from 2 molecule of pyruvates here. Yeah, and from lactate or carbon dioxide and ethanol, or it can go into oxidative phosphorylation, okay, and produce more ATP, 30 ATPs here. So glycolysis pathways involve 10 reaction or 10 steps. Okay, first is the phosphorylation. Phosphorylation of glucose to give glucose 6-phosphate, the isomerization of glucose 6-phosphate to give fructose 6-phosphate. Okay, from the names here, you must know that what happened at each stages. So here phosphorylation meaning there's a addition of uh, there's a phosphorylations going on. Okay, isomerizations here means it's changed or converted into the isomer. So glucose 6-phosphate uh, is converted to fructose 6-phosphate, the isomer of which is a isomer of glucose 6-phosphate. And then again, third steps is phosphorylation of fructose 6-phosphate to produce fructose 1,6 biphosphate. Okay? And then cleavage of fructose 1,6 biphosphate to give glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So here is the second stage of glycolysis. All right. For the fifth steps is the isomerization of dihydroxyacetone phosphate to give glyceraldehyde 3-phosphate. 6. The oxidation of glyceraldehyde 3-phosphate to give 1,3-bisphosphoglyceride. 7 steps. Steps number 7 is the transfer of a phosphate group. Okay, from 1,3-biphosphoglyceride glycerate to ADP to give 3-phosphoglycerate, and the isomerization of 3-phosphoglycerate to give 2-phosphoglycerate. Steps number 9 is dehydration of 2-phosphoglycerate to give phosphoenolpyruvate, and the final step is the transfer of phosphate group from phosphoenolpyruvate to ADP to give uh, final products pyruvate. So for the first 5 fractions of steps, this is what we call as the phase 1 or first stage. And the following 5 fractions is phase 2 or second stage. So phase 1 is basically the investment phase, which you uh, use ATP. Okay, the second stage is where you can produce this, where glycolysis will produce ATP. So the first 5 steps regarded as investment phase since they consume energy. Okay, to convert glucose into 3 carbon sugar phosphates, which is glyceraldehyde 3-phosphate, or also known as G3P. Okay, and then in this intermediate reactions to produce glyceraldehyde 3-phosphate, okay, you need 2 ATP molecule. So this is why we call it as investment phase. So for the first 5 reaction, we are going to use, this pathways is going to use 2 ATP molecules. And the second stage of glycolysis, as it is known as payoff phase, okay, it is characterized by a net gain of ATP and NADH. So even though it has used ATP in the earlier stage, okay, for the second stage, it can produce more ATP and can pay off the ATP that have been used in the first stage. Okay, remember that glucose has 6 carbon, and towards the end of the first stage, you can produce 2 molecule of 3 carbon. Okay, and this is what we call as triose sugar. And then this reaction, okay, happens or occur twice for 1 glucose molecule. Okay, and this second stage, it will produce 2 NADH molecule, 4 ATP molecules. Okay, so with 4 ATP molecules, remember that the first stage use 2 ATP. So it's leading to a net gain of 2 NADH molecule and 2 ATP molecule for 1 molecule of glucose. Okay, conversion of glucose to glyceraldehyde 3-phosphate happen in cytoplasm. So in step 1 of glycolysis, glucose is phosphorylated, meaning the additions of phosphate group. So it phosphorylated to give glucose 6-phosphate. And this is a control point. But this cannot be the only control point since glucose can enter glycolysis pathways as glucose 6-phosphate. Okay, this reaction is endogenic, meaning it needs energy. So where does this energy comes from? It is from the hydrolysis of ATP. So this is a step where ATP being used. And the enzyme involved in the first step of glycolysis is kinase. So kinase is a class of enzyme that transfer phosphate group from the ATP to a substrate. substrate here is glucose. So how this enzyme works is, this enzyme, it has two domains, okay, meaning two parts. So when glucose is attached to this enzyme, okay, water will be removed from the complex to tighten the complex, okay, and this can hydrolyze ATP to ADP. So here is a glucose molecule, okay, you need ATP, okay, and you need enzyme hexokinase and produce glucose 6 phosphate. So glucose itself is a non-charged molecule, but glucose 6 phosphate is a charged molecule, okay, because you have one additional phosphate group. So non-charged molecule to charged molecule, which is a polar molecule. So this uh, the presence of glucose 6 phosphate, uh, it changed the conformation of glucose. So this molecule is trapped inside the cell, okay, because it cannot cross the membrane. Remember, it happened, this step 1 of glycolysis, it happened in cytoplasm. So we need to make sure glucose stays in the cell. So how they do that is by changing the conformation of glucose to glucose 6 phosphate, which is a polar group, polar molecule. Make it polar, so it cannot go outside or it cannot cross the cytoplasm. So this glucose 6 phosphate, the conversion of glucose to glucose 6 phosphate, it destabilizes the structure of glucose. So when it destabilizes the structure, meaning it has high energy. When it has high energy means it is more active. So remember that glucose 6 phosphate is highly reactive, high energy, more reactive, and this is not a stable molecule. So in the second steps of glycolysis is the isomerization of glucose 6 phosphate to fructose 6 phosphate. Okay. So glucose 6 phosphate basically it is reduced to a hydroxy group. So okay, why we need to isomerize glucose 6 phosphate to fructose 6 phosphate? Remember, we want to break down this glucose molecule. Glucose, it has 6 carbon molecule. So we want to make it symmetry. 3 carbon and 3 carbon. 2 3 carbon molecule. So glucose 6 phosphate is not symmetry. But fructose 6 phosphate is symmetry. So that is why we isomerize glucose 6 phosphate to fructose 6 phosphate. Okay, this is the difference between glucose 6 phosphate and fructose 6 phosphate. Okay, glucose 6 phosphate is aldehyde. Okay, this is aldehyde, and fructose 6 phosphate is ketone. Okay, this is chemical groups. And in step 3 is fructose 6 phosphate. Okay, fructose 6 phosphate is then phosphorylated to generate fructose 1,6 biphosphate, meaning here phosphorylated, you add another phosphate group. Okay, so fructose 6 phosphate at carbon number 6, you have phosphate group. So when you phosphorylate it, okay, using phosphofructokinase, you add one more phosphate group at carbon number 1. Okay. Okay, unlike glucose 6 phosphate and fructose 6 phosphate, this fructose 1,6 biphosphate has no other pathways. So it has to undergo the rest of glycolytic reactions. No choice for this molecule. Okay, so this fructose 1,6 biphosphate is highly charged, unstable, and highly reactive molecule due to the phosphorylation reaction. Remember in the first steps of glycolysis, okay, where glucose being converted to glucose 6 phosphate, okay, we add charge. When phosphate groups is being added, it is a charged molecule and it is reactive. Okay, here we add another phosphate group, so it is more reactive and more unstable and highly charged due to the charge charge molecule of phosphate. So what is phosphofructokinase? uh, PFK. Okay, this is a key regulatory enzyme in glycolysis. So, so this PFK is a tetramer. Okay, tetramer, and it has this allosteric feedback. Okay, this PFK, this phosphoenolpyruvate, which the product of steps number 9, it binds to this enzyme PFK. Okay, when the end product of steps number 9 bind to PFK, the PFK and the conformation of PFK change. Okay, so when the conformation change, it destabilizes the binding of fructose 6 phosphate. Okay, and thereby diminishing the catalytic activity. Okay, here you need to know what is allosteric feedback. You have learned this, I suppose you have learned this in uh, enzyme reaction. Huh? Okay, so when there's more of the product, you will get less of the reaction to produce the particular product. So here, if phosphor, sorry, phosphoenolpyruvate is abundant, okay, so it will bind to this enzyme. When it binds, it will change the conformation. When the conformation change, it cannot bind to fructose 6 phosphate. When this enzyme cannot bind to fructose 6 phosphate, meaning steps number this previous steps number 3 cannot happen or cannot occur because there's no enzyme to bind to fructose uh, 6 phosphate. Okay? So you cannot produce fructose 1,6 biphosphate. So if less of product of step 9, which is phosphoenolpyruvate, and so the enzyme is free, enzyme is in its initial conformation, then it can bind to fructose 6 phosphate and producing fructose 1,6 biphosphate. Okay, so it depends on the how much of phosphoenolpyruvate is being produced. Okay, and it will decide if glycolysis uh, reactions, okay, if glycolysis needs to be continue or not. Okay, so when the pathway is producing plenty of phosphoenolpyruvate and feedback inhibitor. Remember what is feedback inhibitor? Okay, to slow down the pathways and decrease the rate of reaction catalyzed by PFK. Okay, meaning it slow down steps number 3. Okay, besides product of step numbers 9, which is PP, ATP is also an allosteric effector. Okay, high level of ATP will inhibit the enzyme. Okay, and low level of ATP will activate. Okay, and also fructose 1,6 biphosphate is an allosteric effector. So you have to really understand about this enzyme. Okay, it is highly affected by the amount of ATP, amount of fructose 1,6 biphosphate, as well as the amount of end product of step number 9, which is phosphoenolpyruvate, or also known as PEP. Okay, so how much of ATP can it activate this enzyme, or can it deactivate the enzyme? Okay, how much of PEP, high amount of PEP, can it activate or deactivate the enzyme, PFK enzyme? So you need to really understand about this PFK. Okay, so PFK is so steps number 3 is basically a control point because of PFK. Okay, because of this enzyme, which can be regulated by the amount of ATP, amount of fructose 1,6 biphosphate, and the amount of PP. So it can control either it wants to continue with the glycolysis, or it can reduce the rates of fraction of glycolysis. Okay, steps number 4 is fructose is where fructose 1,6 biphosphates can be split into two carbon, which is uh, DHAP, dihydroxyacetone phosphate, and diglyceraldehyde 3 phosphate. Okay, so this is the reverse of aldol, which is alcohol-aldehyde condensation, and catalyzed by aldolase. Because it is aldol condensation, so the name of the enzyme is aldolase. Okay, in step number 5, this DHAP, okay, is converted to glyceraldehyde 3 phosphate. Okay, before that, for steps number 4, okay, fructose is being converted, is being split into two, which is the DHAP and the glyceraldehyde 3 phosphate. Okay, each has 3 carbon. DHAP has 3 carbon, and the glyceraldehyde 3 phosphate also has 3 carbon. Okay, okay. In steps number 5, this DHAP is being converted to diglyceraldehyde 3 phosphate. Okay, because DHAP, it cannot continue the glycolysis pathways in the second stage. Okay, it is diglyceraldehyde 3 phosphate that continue the pathways. So this uh, glyceraldehyde 3 phosphate, okay, is important to continue the pathways. So this is what we call as gap, and this is DHAP. So basically in the 4 steps, okay, so it produce basically 4% of sorry, fructose 1,6 biphosphate, it produce like 4% of gap, and the rest it produce DHAP. Okay, So gap lies directly on the glycolytic, directly go to stage number 3 of glycolysis without changes in structure, meaning it can go straight to continue the glycolysis pathways. Uh, and for DHAP, must be modified to continue glycolysis. Okay, however, you must understand that fructose 1,6 biphosphate mostly produced or mostly converted to mostly being split into DHAP. Okay, about 96%. Okay. So less of GAP is being uh, produced. Okay. So both both compound, they are trioses. Okay. So this is DHAP molecule. Where's my cursor? So this is DHAP molecule. Okay. You can see that this is the important part. Movement from carbon number 1, okay, to carbon number 2. Okay, this is carbon number 1 of DHAP. Okay, with this triose isomerase, okay, it move hydrogen from carbon number 1 to carbon number 2 and produce diglyceraldehyde triphosphate. Okay, this is the first stage of glycolysis. First 5 steps of glycolysis in the first stage. Okay, for the first steps, of glycolysis, this reaction is irreversible. Yeah, this is the first steps. This is the second steps and the third step. Third step is also irreversible. Okay. So for the first stage, you have 2 irreversible steps in step number 1 and step number 3. Okay, for the first stages of glycolysis, glucose is converted to 2 molecules of glyceraldehyde triphosphate. Okay, the key intermediate in this series of reaction is fructose 1,6 biphosphate, because towards the end it is produced fructose 1,6 biphosphate. This is the enzyme that catalyze this reaction is phosphofructokinase, okay, which is subject to allosteric control. So this is the first stage, which is a investment stage. Okay, for the second stage, start with steps number 6 is the oxidation of glyceraldehyde triphosphate to 1,3 biphosphoglycerate. Okay, it involves 2 reactions. Okay, addition of phosphate group and together with electron transfer, and when NAD plus is reduced to NADH. Okay, so meaning here hydrogen molecule from NAD, meaning here uh, hydrogen from carbon number 1. Okay, and then when we have these phosphate groups is added to carbon number 1. Yeah, it has one phosphate group here. Okay, since it is the oxidation of glyceraldehyde triphosphate to 1,3 biphosphoglyceraldehyde. Okay, There's another phosphate group at carbon number 3. Okay, 1, 3. 1, carbon number 1 has one phosphate group, and carbon number 3 has another phosphate group. Okay, so towards the end, this is the reaction glyceraldehyde 3-phosphate, okay, plus NAD plus one uh, phosphate group, okay, to produce 1,3 biphosphoglyceraldehyde with two phosphate group plus NADH plus H plus. Okay, NAD plus being reduced to NADH plus one electron transfer. So here is the two reaction of steps number 6. Okay, because it has two phosphate groups added to the molecule. Okay, and this NAD plus with the helps of water molecule H2O, okay, at carbon number 3. Okay, it add one phosphate molecule at carbon number 3. Okay, from glyceraldehyde 3-phosphate to 3-phosphoglycerate plus NADH plus 2H plus. Okay, second reaction here is the phosphorylation reaction where 3-phosphoglycerate okay, plus phosphate group plus H plus, okay, to produce 1,3-biphosphoglycerate plus H2O. For step number 7 is a conversion of 1,3 bisphosphoglycerate to 3-phosphoglycerate. And this step involves another reaction in which ATP is produced by phosphorylation of ADP. And 1,3-biphosphoglycerate transfer phosphate group to ADP. And this is known as substrate level phosphorylation. And this reaction is being catalyzed by phosphoglycerate kinase. So you must know that 1,3-bisphosphoglycerate, it has higher phosphor potential, okay, to release ATP, and this potential of releasing ATP is much more higher than ATP itself. Okay, and here 1,3-bisphosphoglycerate, since it has higher potential to release phosphate group than ATP. So whenever ADP is present, it has it wants to give the phosphate group to ADP. Okay, since it has higher potential than higher potential of giving phosphate group than ATP, this is why it is so important in the step number 6 to produce 1,3-bisphosphoglycerate. Alright, here the reaction, total reaction at step number 7 is 1,3-bisphosphoglycerate with 2 phosphate group here. And when ADP is available, it will gives one phosphate group, okay, from carbon number 1 here to ADP, okay, and this this reaction being catalyzed by phosphoglycerate kinase. And step number 8 is the isomerization of 3-phosphoglycerate to 2-phosphoglycerate, which is catalyzed by phosphoglyceromutase. Okay, why we want to isomerize from 3-phosphoglycerate to 2-phosphoglycerate? Because of the addition of charge group at carbon here. So here, carbon number 3, okay, the charge at carbon number 3 and the charge at carbon number one is the distance is higher. Okay, compared to when we transfer the group, okay, the group to carbon number two. So the charge between carbon number two and carbon number one is nearer. When it is nearer, it gives you higher, uh, it, it is much more unstable. Okay, when it is much more unstable, okay, it has a potential to um, convert to other compound, other molecule easily. So here in the name of phosphoglyceromutase, mutase is the enzyme class of enzyme that move one group. Here is phosphate group. Okay, the removing of phosphate group from molecule of phosphoglycerate. Okay, that is the function of mutase. I repeat, the mutase is an enzyme that transport or that remove one group. Here in this case is phosphate group, okay, from molecule of phosphoglycerate. Okay, it transfer from carbon number 3 to carbon number 2. Okay, so this is why the name of this enzyme is phosphoglyceromutase. For step number 9, okay, so these two phosphoglycerate loses one molecule of water, producing phosphoenolpyruvate (PEP). So enolase is the enzyme that catalyzes the reaction, and this require magnesium 2 plus as a cofactor. And phosphoenolpyruvate (PEP), it has high energy bond. Okay. And step number 10 is the uh, is when phosphoenolpyruvate transfer its phosphate group to ADP, producing ATP and pyruvate. And this is one of other control point in the glycolysis. And this reaction is catalyzed by pyruvate kinase. Overall, this is the second phase of glycolysis. Okay. The second stage of glycolysis, where we call it as a payoff, where we produce 4 ATP, okay, and pay back the use of 2 ATP at the earlier stage. Okay, the production of ATP happen at these steps, okay, and happen at this step, 2 steps. Production of ATP happen when at 2 steps here. Remember that here from steps number 6 to steps number 10, 2 molecule of GAMP, okay, glyceraldehyde 3-phosphate. Okay, glyceraldehyde 3-phosphate, 2 molecule of glyceride 3-phosphate. Remember, 6 carbon glucose, okay, they are converted to towards the end of steps number 5. Okay, they are converted into glyceraldehyde 3-phosphate and uh, DHAP. So DHAP is a isomer of GAP here, glyceraldehyde 3-phosphate. Okay, since glyceraldehyde 3-phosphate lies in the direct pathways of glycolysis, so glyceraldehyde 3-phosphate will undergo the glycolysis, but not DHAP, since DHAP cannot continue the glycolysis, as unless it's being converted to its isomer glyceraldehyde triphosphate. Okay, at step number 5. So here after step number 5 here, okay, and step number 6 here, you can produce ATP. So this is 2 ATP, and here another 2 ATP. So total here 4 ATP. So 4 ATP is being produced at second stage of glycolysis, and 2 ADP being used at the first stage of glycolysis. So at the end, you can get a summary of 2 ATP net. The control point in glycolysis, there's being catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase. So here I will just summarize everything. So in step one of glycolysis, when glucose being converted to glucose 6-phosphate, okay, it makes the structure of glucose more reactive and high energy, okay, due to the additional of phosphate group at step number 1. This step also prevent glucose from crossing the membrane. Remember that glycolysis happen in cytoplasm. So in order to prevent glucose to cross the cell membrane, okay, glucose needs to change its conformation. Okay, how they've been doing this is by add the phosphate group to glucose. Besides making it highly reactive, highly unstable, high in energy, it also changed the conformations to prevent it from crossing the cell membrane. Okay, at steps number 1, it is control point, okay, which inhibited by its substrate, which is glucose 6-phosphate. Second control point would be at step number 3, in which fructose 6-phosphate is being converted to fructose 1,6-phosphate biphosphate, and these steps is inhibited by ATP. The amount of ATP produced towards the end of glycolysis at stage 2 glycolysis. Okay, if too much of ATP, it will attach to the enzyme at at steps number 3 here. Okay, when it combined to the enzyme, it changed the conformation. When it changed the conformation, fructose 6-phosphate can no longer attach to that enzyme, and therefore cannot be converted to fructose 1,6-biphosphate. So this act as control point number 2. Okay, so note that 2 control point happen at the first stage of glycolysis, okay, which is the investment stage. Okay, and then at the second stage, the last control point happen at the last steps of glycolysis, when PP, phosphoinopyruvate, is being converted to pyruvate. This is also inhibited by ATP. Okay, too much ATP or too many ATP will attach to phosphoinopyruvate, prevent it from uh, converting to pyruvate. So this is 3 total control point in glycolysis pathways. I would like also to highlight that at the first stage of glycolysis, basically the molecule are being unstabilized. Okay, they want these pathways. It makes the glucose molecule unstable, so it has high reaction, highly unstable, so it wants to change its conformation faster. Okay, meaning it wants to go to the next steps. Okay, they can't wait to change its conformation because they are highly unstable. Okay, but then for the second stage of glycolysis, the one that this molecule in this second stage of glycolysis, basically they want them to be prepared to uh, give phosphate group. Yeah, meaning they want to produce ATP. Okay, so this molecule, they are being prepared. Okay, whatever happen at each steps of the glycolysis, they are being prepared so they have higher energy, higher potential than ATP to give the phosphate group attached to them to ADP. So ATP can be produced from ADP. Okay, so we have to know what happened at stage 1. What is the purpose of stage 1 of glycolysis? Basically, they want to prepare the molecule to be highly reactive so it can move to other reaction faster. Okay, and for the second stage of glycolysis, the molecule change so that the molecule itself has higher potential to give phosphate group, okay, than ATP. Okay, when it has higher potential of giving phosphate group than ATP, whenever ADP is available, they want to give that phosphate group to that ADP to produce ATP. Okay, so basically this is what happened at stage 1 and stage 2 of glycolysis. So to summarize everything, in the final stage of glycolysis, two molecules of pyruvate are produced for each molecule of glucose that enter the pathways. Okay, remember, two molecule of glucose. Okay, sorry, one molecule of glucose producing two molecule of GAP, GAP. Okay, one each for DHAP and one molecule of GAP, which DHAP can be later converted to its isomer GAP. Okay, this reaction involve electron transfer as well as the net production of 2 ATP for each glucose and 3 control points in the glycolytic pathways. So glucose, it can be metabolized with or without oxygen. One is in aerobic metabolism, which is in the presence of oxygen, and another is anaerobic metabolism, which happen in the absence of oxygen. Basically, this happens in cytoplasm of cell. Okay, the metabolism of pyruvate. What happened at the end of glycolysis? Okay, glycolysis produce pyruvate. So glucose, during glycolysis, glucose produce two molecule of pyruvate. Okay, electrons and two molecule of ATP net. So several metabolic fates are possible for pyruvate. In aerobic metabolism, which when oxygen is available, pyruvate, it lost carbon dioxide and the remaining two carbon atom link to coenzyme A, producing acetic group to form acetyl-CoA. Okay, this acetyl-CoA can enter the TCA cycle. In other words, aerobic metabolism of pyruvate, it involves the second, third, and fourth stages of carbohydrate metabolism, okay, which I mentioned earlier, which the formation of acetyl-CoA, TCA cycle, and ETC, electron transport chain. When in anaerobic metabolism, when oxygen is unavailable, what happened to pyruvate is it will be capable of alcoholic fermentation. When pyruvate lost carbon dioxide, okay, similar with where in aerobic metabolism, okay, when the pyruvate loses carbon dioxide, it produces acetaldehyde, which can be reduced to produce ethanol. So basically acetaldehyde is unstable conformation. Okay, so it will convert to whatever conformation that make it stable, which is ethanol. So here anaerobic metabolism of pyruvate in organism. So this pyruvate, where's my cursor? So this pyruvate, okay, is being converted to acetaldehyde and ethanol. So acetaldehyde, okay, by the use of enzyme pyruvate decarboxylase in the presence of magnesium 2 plus, Mg2 plus as a cofactor, okay, and TPP, pyrophosphate. And second is ethanol by alcohol dehydrogenase. So this is how pyruvate is being converted to acetaldehyde by pyruvate dicarboxylase. It removes the carbon. Okay. So here it has this carbon double bond O. Okay. At carbon number 1 also has a double bond, and carbon number 2 also has one double bond. Okay, and then it move it move carbon dioxide from this carbon. Okay, and becomes it release carbon dioxide and producing acetaldehyde. Okay, this acetaldehyde, okay, is further converted to ethanol by alcohol dehydrogenase. So two reactions leads to the production of ethanol. One is decarboxylation of pyruvate to acetaldehyde, and second is the reduction of acetaldehyde to ethanol. Okay, pyruvate decarboxylase is the enzyme that catalyzes the first reaction here. And it requires Mg2 plus and cofactor TPP. And second, in the second reaction, it uses alcohol dehydrogenase, which catalyzes the conversion of acetaldehyde to ethanol. So basically, in anaerobic metabolism, besides producing ethanol, okay, in other organisms, pyruvate is being reduced to lactate by lactate dehydrogenase. So pyruvate plus NADH plus H plus producing lactate plus NAD plus. So what is lactate? Lactate is a compound produced from pyruvate when glucose is metabolized in the absence of oxygen. So basically during intense exercise, lactate can be accumulated in the muscle. So the intracellular pH in the muscle will be dropped, and sometimes when we feel cramps, that is what happened. Okay, when the pH drops. So this lactate in the muscle, eventually it can be diffused into the bloodstream and take up by the liver to produce glucose. So conversion of lactate to pyruvate by lactate dehydrogenase or pyruvate to lactate, okay, by NADH plus H plus depends on the intracell concentration of pyruvate and lactate and the ratio of NADH over NAD plus in the cell. So basically, this is how they control the amount of glucose. When glucose is lower, okay, and lactate is and it produces lots of lactate, yeah, high content of lactate, okay, in the blood, and liver will take up that lactate, and then lactate would be converted to glucose to produce glucose to supply the ATP.
In the liver and heart, the ratio of NADH over NAD plus is lower than exercising muscle. Yeah, because exercising muscle produce a lot of lactate compared to liver and heart. So this tissue in liver and heart, okay, which obtain from the blood, it can oxidize lactate to pyruvate. Okay, and then in the liver, pyruvate is converted to glucose by gluconeogenesis or oxidized in the TCA cycle. It depends. In heart muscle, they use to oxidize lactate to carbon dioxide and water by using TCA pathways. So this is happen only in heart muscle. In the liver, it can choose either to use gluconeogenesis to produce glucose or oxidize in the TCA cycle, meaning continue with the glycolysis pathways. So, to summarize everything, in anaerobic metabolism, under anaerobic condition, the most important pathway for regeneration of NAD plus is the reduction of pyruvate to lactate. And the enzyme lactate dehydrogenase LDH is a tetrameric isoenzyme, okay, which consists of different subunit H and M, okay, which H4 predominates in heart and M4 in skeletal muscle because heart has different function than muscle. That is why although it has the same lactate dehydrogenase, it contain H and M subunits, two different subunits.